Hello, my name is Raymond Blind. I'm an assistant professor at Vanderbilt University School of Medicine in the Division of Diabetes, Endocrinology, and Metabolism, with appointments in the Department of Medicine, Biochemistry, and Pharmacology. The title of what I'm going to talk to you about today is Structure-Based Anti-Diabetic Therapeutics. And what the talk is really centered around is what this image you're looking at from a publication we had a few years ago that is based on a nuclear receptor being regulated by a particular type of phospholipid that is really important in signal transduction in mammalian cells. So the problem that we work on in the lab is fatty liver, and it is actually a huge problem, particularly in the United States. There are some recent epidemiological studies that have been done that show that about 60 million Americans actually are afflicted with fatty liver to some degree or another, and 70% of type 2 diabetics have this fatty liver disease. Particularly in Tennessee, where Vanderbilt is located, 15% of the total population, or 820,000 people, have diabetes. So fatty liver itself is actually a huge problem for Tennessee and Nashville. So what does fatty liver do? It's actually induced by diet often. Particularly, it correlates with type 2 diabetes. And what this does is it causes a buildup of particular phospholipids and cholesterol bodies that accumulate in the liver. If that can often lead to pathologies in the liver that result in a decrease in the liver function. But when coupled with other challenges faced by the liver, particularly in diabetes, the liver can begin to lose function. And that is a huge problem in diabetic liver pathology in particular. So what does the liver do that it matters that it loses function? Well, it does a lot of things. It's an extremely important organ. However, one of the things it does is use cholesterol and phospholipids to make bile or bile acids. And that process is actually controlled by a particular protein that binds DNA called the transcription factor, which is named NR5A2. And what that transcription factor does is it activates genes that will actually allow the liver to accumulate phospholipids and cholesterol into the bile duct that you can see. That's the green organ right there. Or I'm sorry, through the bile duct into the gallbladder. And the gallbladder stores that bile until you eat a fatty meal. And when you eat the fatty meal, the bile duct will release the bile into the bile duct, and you'll get a detergent effect that will allow your body to absorb all of these really nutritious phospholipids and other cholesterol type of molecules that you're taking up in your diet. So this is one of the functions that the liver performs, and that is controlled by this NR5A2 transcription factor. So NR5A2 is an important member of the nuclear receptor superfamily. So these are all transcription factors that are regulated by small molecule ligands that they bind to. And these proteins have a DNA binding domain and also a domain that allows them to interact with their hydrophobic ligands. Most people are familiar with the estrogen receptor, which is a member of this superfamily of transcription factors, which is often dysfunctional or hyperactivated in breast cancer. Also, the glucocorticoid receptor is an extremely important member of this family, but we study the NR5A members of this family, which are highlighted in this diagram here as SF1 and LRH1, but I'll refer to them just as the NR5A family. And essentially, there was a recent publication that came out from a different group, David Moore's group at Baylor, that showed that a particular component, a phospholipid component of soybeans, can actually alleviate a lot of diabetic effects in the liver. And that component of the soybeans is called dilauryl phosphatidylcholine. So that's a very long word, but I'll just call it DLPC for short. And this was really a remarkable paper that they published, and it was able to show, as you can see in these diagrams here, that when they fed mice this DLPC phospholipid, they actually saw a dramatic decrease in glucose homeostasis in these mice. So glucose homeostasis is just the ability of your body to regulate blood sugar. So by this, on the upper panel, you see a glucose tolerance test, and on the lower panel, you see an insulin tolerance test. And the red line is the DLPC-treated animals, and the black line is just a vehicle control. And you can see that there are decreases in the red line versus the black lines. And that is essentially an improvement in the diabetes that's afflicted upon these animals in the form of a diet-induced obese state. So it's more reflective of type 2 diabetes than type 1 diabetes. So also what they found was that these effects were completely dependent on this nuclear receptor called NR5A2 that I've been talking about. So you can see on the far panel actually that in mice that lack that NR5A2 in the liver, they completely loses the ability of DLPC to modulate the glucose homeostasis in those mice. So this is really dramatic and very quantitative example of how this DLPC phospholipid can regulate glucose homeostasis in the mice. But just looking at a more descriptive picture, you can see here, again, in these same mice, they were able to induce fatty liver in the mice, which was completely alleviated by the DLPC treatment in the upper panel. So these are just two different types of stain, which you can see are causing a decrease in the white spots or the red spots upon DLPC treatment, which represent fat deposits in the liver. But on the further away from me panel, you could see that the NR5A2 knockout animals were completely unable to respond to that DLPC phospholipid. So essentially, these data all demonstrate that DLPC is able to alleviate fatty liver and lots of the diabetes associated with glucose homeostasis effects in mice. And all of this is operating through the NR5A2 nuclear receptor. So this makes it extremely important to understand how DLPC is actually activating this particular nuclear receptor called NR5A2. So that's what my lab studies. We take crystallography approaches, very high resolution quantitative approaches to understanding how phospholipids binding to nuclear proteins can actually regulate their function. So how does this generally work? So we have this NR5A transcription factor, and as I'm showing you here, we have crystal structures of it bound to different phospholipids. And one of the most important discoveries that we have made is that this NR5A group of transcription factors is regulated by signaling phospholipids. So those are very important phospholipids that are involved in signal transduction at the membrane, and I'm going to talk about these as PIP2 and PIP3, as you can see highlighted in that red box there. So what normally do these particular phospholipids actually do in normal cellular physiology? And that is that they form docking surfaces at biological membranes. So essentially what these phospholipids do is they are generated on the cellular membrane in response to some sort of signal, such as insulin, that comes in from the outside of the cell. And as you can see here, this PI345P3, that is the PIP3 phospholipid, that forms a little docking site for that brown and green protein to actually bind to and get clustered onto the membrane. And by clustering onto the membrane, those proteins are more frequently able to bump into each other. And because of that, you can get an activation of those proteins. And so PIP3 in these membranes is actually very well known to cause this type of clustering phenomenon among proteins onto membranes. So that's why this PIP3 phospholipid is so important. And so we investigated that phenomenon for quite a while, how PIP2 and PIP3 might regulate the NR5A transcription factors, and we were able to come to the conclusion that PIP2 is able to inhibit transcription from NR5As, and PIP3 activates transcription from these NR5A transcription factors. But what we went on to do after that publication was ask the question of how does PIP3 and other phospholipids such as DLPC, how do they activate NR5A transcription factors at atomic resolution with looking at it from a molecular point of view. And so in order to really address that question, you have to have a little bit of background about how nuclear receptors generally are activated. And it's very well established that nuclear receptor ligands are able to activate the nuclear receptor through a mechanism called allostery or shape change. So essentially what happens is that the the nuclear receptor is in a molecular inactive position. And when it binds to an activator, it will change shape. It'll physically change shape and wrap itself around that small molecule activator. And that shape change opens up a novel activation surface on the nuclear receptor. So the nuclear receptor shifts from this active conformation that's shown in red to an inactive conformation that's shown in blue. And that's really driven by the shape change that's occurring, that's being driven essentially by the ligand binding to the nuclear receptor. So what that shape change does, like I said, is open up an active interaction surface for a transcriptional co-regulator to come along and bind to. So this allosteric switch mechanism is very well established to be how ligands that bind nuclear receptors can turn on and turn off the nuclear receptor transcriptional activity and DNA binding activity. So what we wanted to do was just basically ask the question, how do the inhibitor, which is PIP2, this phospholipid, and the activator, PIP3, do they induce any shape changes in the nuclear receptor that we study, NR5As? So we investigated this and were able to show that NR5A bound to PIP3 or NR5A bound to PIP2 are both in conformationally identical states. So in other words, there is no shape change when we bind NR5A to PIP2 or to PIP3, which is very strange. We were very surprised by that result because, like I said, all nuclear receptors are known to be regulated by allostery. We also did this with the other member of the nuclear receptor, the NR5A2 nuclear receptor, and we were able to also show that there aren't any huge significant conformational changes that occur in the NR5As when they're bound to any phospholipid whatsoever. Although there are some slight changes in the function of these nuclear receptors when they're actually bound to different phospholipids. But given the dramatic effects that DLPC and other phospholipids, signaling phospholipids, can have on NR5A activity in mice and in cellular systems, we don't view the changes that occur as being really strongly significant. So when the data don't really match what your model states, you often have to change the model of how your nuclear receptor works. So we've done that, and instead of relying on conformational change for these phospholipids to bind to and regulate the NR5A nuclear receptors, we actually think the data are more consistent with a model where the phospholipid head group actually forms a novel docking surface on the NR5A transcription factor. So like I said in the beginning of this talk, the signaling phospholipids are well known to operate that way at membranes. You can see here the PIP phospholipid is able to bind to a protein called protein A, and then we're actually hypothesizing that the same thing happens with the NR5A2 transcription factors, just with a different subset of proteins perhaps, and that there are actual proteins that will bind to this NR5A2 transcription factor through the phospholipid head group. So we've done some discovery proteomics experiments and been able to show that in fact we do find that particular domain-containing proteins can specifically interact with NR5A when it's bound to one phospholipid but not to another phospholipid. So despite the fact that these phospholipids aren't inducing any conformational changes or shape changes in the proteins, we're still seeing dramatic differences in what proteins those two different phospholipid-bound NR5A molecules are able to bind to. And so our hypothesis actually, which is very different from other groups' hypothesis about how DLPC actually works, is that it is not a conformational changer of the NR5A nuclear receptor. But rather than changing the conformation, it is actually competing with endogenous phospholipids to displace the endogenous phospholipid away from NR5A and thus activate the receptor. So of course, if you're displacing that away, it would have to be an inhibitory phospholipid. But as I stated earlier, we know that the PIP2 phospholipid actually does inhibit the activity of the NR5A nuclear receptor. So this is work that's ongoing in the lab right now, and we're characterizing these phospholipid interaction domains and determining if they actually do change NR5A transcriptional activity in human liver cells, and also if these phospholipid binding domains can actually independently inhibit the activity of the receptor when they're decoupled from the rest of their proteins. So this is another thing that these strongly suggest is that the way that industry and pharmaceutical companies have been screening for agonists of the NR5A2 nuclear receptor may have been proceeding without all the information that they needed to actually accurately drug this receptor. So many pharmaceutical companies for decades have been trying to find small molecule activators of the NR5A subclass of nuclear receptors without much luck whatsoever. And that is because a lot of the drug design, the drug screens that they use to look for these molecules have relied on an allosteric model of nuclear receptor regulation. So they were always looking for a molecule that would induce this active conformation away from the inactive conformation. And clearly our structural biology approaches have revealed that there isn't a dramatic shape change in these two nuclear receptors upon binding to different phospholipid ligands. So we are actually initiating a new type of nuclear receptor screen in the lab, initiated by Corey Seacrest, a PhD student in the lab, that actually, rather than looking for allostery regulation of this nuclear receptor, we're just looking for small molecules that were just competitively displace endogenous phospholipids away from the nuclear receptor. And we are going to quantitatively compare this new, different kind of drug screen to classic drug screens that are operating through allostery, and then determine which of those small molecules are actually better agonists of the NR5A nuclear receptors. And this is work that's currently funded by the Vanderbilt Diabetes Center through a Discovery Award, as well as the Vanderbilt Diabetes Research and Training Center. So we're very excited to get that work underway, and like I said, Corey Seacrest is doing those studies right now. So I'd like to thank Al Powers and John York, who are responsible to a large degree for bringing me to Vanderbilt, as well as my postdoc mentor Holly Ingram and co-mentor Robert Flederick. Also, I want to thank the NIDDK, NIGMS, American Cancer Society, and National Cancer Institute for all of their support they've provided, as well as the individuals who've done this actual work in the lab. Mainly the data I've shown has been generated by Merced Malabanan, Mark Crowder, and Corey Seacrest, with the help of others in the lab. And I thank you very much for your attention.